Hello, welcome back for our second lecture. This is going to be the basic concepts of infectious disease. Okay, things we're going to be looking at in this chapter is our relationship to the world around us. And we're going to see how microbes can not only move through the human population, but also use other hosts, other organisms to circulate. So our relationship to those organisms will be important. We'll look at the concepts of pathogenesis with specific infections, and we'll again see our place in the cycle of transmission. We talk about a pathogen, we have to be able to identify what a pathogen is. Again, not all microorganisms are pathogenic in a sense. We have E. coli here. E. coli can have different variants that cause them to be pathogenic. We have natural E. coli within our body, which generates vitamin K, as we heard before. But we can also have E. coli that causes gastrointestinal distress as well. Looking at pathogens, we can have pathogens mixed throughout our healthy microbiome. So our microbiome is all of the organisms throughout our body that are part of us. This also includes what they secrete, so proteins around them and other molecules. We can look at our microbiota as just the microorganisms that we find in our human body. When it comes to pathogenic organisms, they can work their way into our body and really compete for space and nutrients with our own microbiota, with the microorganisms that exist on us. We know that our skin is going to be covered in microorganisms as well as our gastrointestinal tract. So everything from our mouth through our digestive system and out is completely already colonized by microorganisms. But pathogens can get in and they'll compete with these microorganisms that are part of our natural microbiota. And what they need to do is have the ability to have adhesion to the cells around them or to other microorganisms within our body. Okay. When we're looking at pathogens, pathogens come in a wide range of sizes. We even have parasites that are pathogens. We have some that are ectoparasites and some that are endoparasites. So ecto outside, endo within. These pathogens can cause disease in humans and in animals. And we'll look at their pathogenicity, their ability to cause disease. Microorganisms themselves first cause an infection by growing within their host. So they have to adhere and begin to grow. Once they're growing, so meaning they are not just getting larger necessarily, but replicating in numbers from 2 to 4 to 8 to 16, we start to have an infection. Microorganisms can divide every 20 minutes. So an infection can mount very, very quickly. So disease occurs when we actually start having symptoms. So they meet a specific threshold, these microorganisms, before they start causing symptoms that we can actually pick up. We have acute infections where we have symptoms that can come and go, like the common cold. Then we have chronic infections that are going to come on over and over again that we don't necessarily just get rid of, like tuberculosis, for example. We have different types of pathogens. We can categorize this in two ways. We have primary and opportunistic pathogens. Our primary pathogens that we're looking at are ones that are going to be causing disease in a healthy host. So anyone with a normal immune system can be infected by a primary pathogen. While on the other hand, a opportunistic pathogen is one that's going to infect somebody with a lowered immunity. So maybe someone with a immunodeficiency, this could be from lupus to HIV, or it could be someone's age, very young or very old, that makes them at risk to disease. Looking at the virulence of these pathogens, we see that primary pathogens have high virulence, meaning they're very, very strong. Looking at ones that affect, ones that are opportunistic pathogens that affect people with compromised immune systems, those are going to have a lowered virulence here. Some of these pathogens also have latent states, and depending on the person's immune system, latency can be the suppression of a disease where we don't see it, or it can be the expression of a disease more frequently if someone's immune system can't control that latent state. Here we see an example of HSV. This is the herpes simplex virus. Here we see that's showing cold sores on the mouth. HSV can be oral or can be genital, found in 2 locations. HSV itself can have a latent state, meaning that while you can have the infection, you have the virus, it doesn't mean that you see this physical manifestation into a cold sore, as seen here. So it can be in a latent state. With all HSV, approximately 70 to 90% of people have an HSV infection, whether that's HSV1 or HSV2, oral or genital infections. But many people actually don't show the signs and symptoms of HSV infections. And that's because it's in a latent state. So the virus is actually storing its genetic information within cells and not actively being expressed. In a latent state, if we wanted to culture something, let's say a microorganism, like even a bacteria that was in a latent state, we wouldn't be able to find it even in a culture. Second, lead to something like a false negative. Talking about virulence, we want to talk about the level of harm a pathogen can cause from an infection. If we go back to this slide, primary and opportunistic pathogens, we saw that the primary pathogens had high virulence, opportunist had low virulence. So that's, again, the impact of the actual pathogen. Okay, when we look at virulence, we have a couple different ways to measure it. We have the lethal dose, LD50, and that shows us that 50% of the population has succumbed to the disease. It's lethal. Infectious dose, ID50, is when 50% of the population is infected. This is how we measure part of the virulence. So in this graph that we see here, we see dose, number of organisms administered per animals. So we're increasing the amount of organisms on the x-axis. And on the y-axis, we see percent mortality. Looking at this here, we have 2 different agents. So microorganism agent 1 and microorganism agent 2. Here we're seeing LD, which is the lethal dose at 50%, which is what we're looking at, percent mortality here. And we want to know whether agent 1 or agent 2 is actually more dangerous, more virulent. So we'll give you a second to look at this and decide whether you think it's agent 1 or agent 2. So agent 1 would be more virulent in this situation because it only takes 400 of the dose administered, maybe 400 microorganisms to cause infection versus one is 600 microorganisms or 600 of a dose, a specific dose. So agent 1 is therefore more virulent than agent 2 based on its LD50. Okay. To bring in more vocabulary associated with pathogenesis, we can start with invasion and invasiveness. Microorganisms have to invade our body. They have to compete with other microorganisms in our natural microbiota. So to invade is to enter in, and pathogens can enter in through many different portals within the body, and different pathogens reside in different tissue types within the human body. Invasiveness is the ability of a microorganism, a pathogen, to move through the body to get to that desired tissue of where it needs to be. These examples here, we see microorganisms coming in. In green, we actually see the microorganism Listeria. Listeria is a microorganism you can get from contaminated dairy products like yogurt. And actually, you probably have heard of Listeria outbreaks from time to time. It's actually a microorganism that once it's in, let's say a yogurt, by refrigeration of the yogurt, doesn't prevent the microorganism from growing, so it continues to grow even at those low temperatures and can mount to very, very high numbers, which is here again bringing up the dose, making it more virulent when it's actually consumed. Once it's in the body, it can actually be an intracellular pathogen. So it's invasion, it's portal of entry. It goes into cells and builds what we see in red here, a little tail, and it actually swims or propels its way through cells like a little rocket. It's its actin tail. We'll see Listeria and many other examples coming up. Okay, looking at host range, this is who can be infected by the pathogen. We have a narrow and broad range. So a narrow range would be one species only. So here we see Salmonella, Enterica, Civaro typhi only affects humans, where we have some Salmonella typhimurium, which actually affects not only humans, but also other animals, meaning it can be spread from animals to humans and humans to animals. So a broad host range means more organisms, more species are susceptible, narrow meaning only some. This is also part of cautious postulates. If we don't have a model organism to test a microorganism on, we're not able to study it. So narrow range microorganisms do not work well with cautious postulates. Okay, let's talk about how we can define disease. So disease itself is a state of the body when the normal function of the body, the normal systems of the body are actually disrupted. So this is what disease is initially picked up at, and it's seen as normally signs and symptoms. When we talk about an infectious disease is one that can be transmitted from person to person or from animal to another, basically transmitted from one to the next. So transmissible disease, it's transferred, and that's infectious disease. So that's not something inherited, it's something picked up. So this could be bacterial, viral, or even parasitic in nature. For anybody who is pre-health or allied health, a lot of the things we go over are very applicable to clinical practices. So looking at signs and symptoms are going to be important for understanding which infections someone may have. Looking at signs to start off with, a sign is an observable marker. So it's an objective marker of disease. Where on the other hand, a symptom is subjective. So if somebody said, I don't feel well or this hurts, that would be a symptom. Where on the other hand, a sign is something you can quantify. So you can actually take somebody's temperature, get an actual reading of a temperature. You can visualize an infection like a fluid-filled rash. When we talk about a syndrome, it's actually the combination of signs and symptoms, and that's what help us diagnose disease. It's collective characterization of these. Looking at signs and symptoms, we can also see that how the host responds to a specific disease is an indicator as well. So our response to an infection is actually known as immunopathology. So that's how our immune system interacts with the invading microorganism. In this case, even our own cells can cause damage to our infected cells. So we may kill some of the infected cells. So here we even see a tumor cell, which is what our immune system goes after, or an infected cell by a microorganism. We have cell death. And what we see here is that we're breaking down our own tissues in immunopathology. There's something called sequelae, which is the consequence following a disease that normal tissue function has been disrupted or damaged from our own immune system. So even after the infection is gone, we have this secondary state of the disease where essentially our own immune system has broken down our own tissue in sequelae. Okay, looking at infectious disease, this is a really good chart showing us number of microorganisms and our immune system's response as there's interplay between the two. So we have response and reactions essentially occurring here. Starting out on the x-axis, we have our incubation period. So this is when the microorganism has gotten into the individual and it started to slowly understand its environment and starting to think about its growth cycle or reproductive cycle here in the incubation phase. So it's starting to take the necessary steps to gather the resources that it needs for reproduction to essentially do asexual reproduction and divide in two. Or if it's a virus, we could look at it taking care of its environment, making sure that it's ending up in the right cell type in order to replicate. Then we have the prodromal phase. In the prodromal phase, here we see the replication of the microorganisms. As this is higher, And our immune response is just upped a little. It notices that something is a little bit off, so it's triggered at first. But we see the microorganisms have already surpassed our immune system's response. By the way, in the incubation phase, we do see the immune system is always constantly monitoring our body, and that's how it's able to pick up in the prodromal phase this increase of number of microbes. Following that, we have the illness phase. This is when we meet a specific threshold that microorganisms are actually altering the way our body is interacting with itself and responses. This is where we would see our signs and symptoms at its peak, and we see our immune system, while it's increased, is not overcoming the microorganisms at this phase. Decline phase is when we switch from the number of microorganisms in high and immune system lower to the opposite. Immune system response is high, number of microorganisms are low, so we're declining in the disease. This is where we're starting to see symptoms dissipate as number of microorganisms goes down, immune system can go up, unless those signs and symptoms are associated with the immune response itself. When we look at the convalescence phase, this is this time when our immune system is continuously monitoring to ensure that we did remove those microorganisms. The number has steadily dropped if we look at it from here to here. And long-term phase, microorganisms drop back down and we have surveillance with those microorganisms. So we can see two different bell curves here. If we're looking at number of microbes in the orange color, we increase here at illness phase and drop back down. If we're looking at the immune system response, we see we peak here at the decline phase and, convalescence phase, and then back down. Okay. When we look at infectious disease, we can look to the CDC, the Center of Disease Control, to take note of what's going on as far as morbidity and mortality rates. Let's go ahead and define these terms. So morbidity is associated with the rate of an illness, where mortality is the rate of death. As you can imagine, if a newscaster used the word morbidity or mortality, it's going to change your view on the situation, on the spread of disease. So morbidity is looking at how many people are ill. Mortality is looking at how many people have succumbed to that illness. Center of Disease Control, again, is watching morbidity and mortality and releases weekly reports. And if you're interested, a fun exercise is going to cdc.gov. You can see any kind of outbreaks that are currently happening, and it will show you morbidity and mortality rates of these diseases and the local trends. Looking at transmission of disease, there's many modes of transmission of different microorganisms, and this happens to be because of their tissue types, the tropism of what they're interested in. Here we can see direct contact, so physical contact, whether that's human to human or human to animal. In image B, we see vehicle transmission. So this can be from fomites. Fomites are inanimate objects. It can be something as simple as a doorknob that you touch, or maybe a water fountain or food you consume, or in the air that you breathe. This is vehicle transmission. We have transplacental transmission as well, which is from mother to child. So it moves through into the child. And in example D, we can see a more complex transmission cycle. We can see a reservoir of animals that carry a specific pathogen. And we can see that it can be transmitted through maybe arthropods like mosquitoes or ticks, and then introduced into humans, or other mechanical vectors as we see here. Okay. Looking at the mode of transmission, we can monitor the infectious cycle. So how does this move from person to animal, and so on? One term we can talk about is horizontal transmission, when we go from person to person or person to animal. On the other hand, vertical transmission is going from parent to child. So vertical transmission is an example of transplacental transmission, where horizontal would be a good example of that direct transmission. When we look at modes of transmission, we have direct contact transmission. Direct transmission is from person to person from physical interactions like touching, kissing, or sexual activity. When we look at indirect transmission, this could be airborne transmission. So droplets of saliva can be suspended in the air, and viruses and bacteria can be suspended in the air too. It's not just one-on-one communication with people that transmits airborne. These microorganisms can stay suspended for over 24 hours, for over the weekend in the air, some of them still alive and viable. Microorganisms can also be indirectly transmitted in the air, in the atmosphere as well. We know that microorganisms can move from the Sahara deserts all the way over to the east coast of the United States and down. So microorganisms can be transmitted through the air, also through fomites, inanimate objects, so things we touch around us, maybe like a pencil, our notebook, a laptop. Vehicles are going to be things such as food, water, and air, whereas a vector, on the other hand, is something living like this mosquito or a tick carrying Lyme disease. And actually, when we talk about the most deadly animals are actually mosquitoes because the fact that they carry disease. So those are our vector-borne diseases. Looking at reservoirs are essentially an environment that's harboring a microorganism. So when we go back to this picture here, we see animal reservoirs. So maybe within the forest, the deer has ticks on them, and these ticks are carrying Lyme disease that then spreads to humans. That's a reservoir. So humans can actually act as reservoirs too, as we are natural animals in our environment. But these reservoirs harbor pathogens. They carry pathogens around communities. We also have asymptomatic carriers. Asymptomatic means you don't have any symptoms. So you could be infected with the disease, but have zero signs and symptoms, but you could still spread this disease to someone else. We may be familiar with this with COVID, that we had many asymptomatic carriers. Okay. Let's look at different types of spreads of disease. We have endemics, epidemics, and pandemics. Looking at endemics, this is something that occurs in a very low rate in a community and typically has an animal reservoir. So like we said with Lyme disease. Epidemic, on the other hand, is really the pickup of a disease. So if we look at the time, in a short period of time, all of a sudden we see a large number of cases coming out. Where in endemic, we wouldn't have a large number of cases, a low rate over a long period of time, a well-known disease. Pandemic, on the other hand, which you're familiar with COVID-19, we also have influenza and HIV, is disease that spreads worldwide. There's no barriers that prevent the movement of these diseases. Okay. We also have what we call zoonotic diseases. Zoonotic diseases are diseases that are spread from animals to humans. When we think of many of the diseases that we've heard of, we can think of swine flu from pigs. We can think of bird flu, avian flu from birds. There's a lot of diseases that can be spread from animals that we interact with to humans themselves. Okay, Lyme disease itself is going to be spilling over into humans. So it doesn't actually cause disease within the animal host. So let's say if there is a tick on a deer, the deer is not the tick is actually not going to be affected by the disease, but it can go from the disease reservoir, the deer, into the human population. So when humans go into areas, forested areas, they can come into contact with these ticks and it could spill over into humans and cause disease in humans. Ebola, on the other hand, doesn't only cause disease in humans, but can also cause disease in the host that it's in. Okay. When we talk about animals and zoonotic diseases, animals are essentially what we call incubators for new diseases. We've probably heard that it's really important to make sure that we take our antibiotics to the full extent, which it is true. And we've also heard that we want to make sure we're only taking antibiotics in situations where we have a bacterial infection, not a viral infection. When we look at the use of antibiotics, over 70% of antibiotics are actually used in animal agriculture as animals are kept in small confined areas where disease spreads easily. We know in cities where people are packed in tight, disease spreads easily from person to person as compared to a more suburban area or spread out farm area where people don't have as much close contact. Well, the same thing applies to animal agriculture, where we have these densely packed areas of animals, and disease spreads from animal to animal very quickly. When this happens, farmers normally lose their entire flock of animals that they have to be put down at this point. But what happens is that farmers end up treating with antibiotics these animals, and by pumping them with antibiotics, it prevents them from succumbing disease and spreading disease. So it increases the yield of product, of which is the animal in this case. We can also look at how viruses spread within these animals. So two viruses, because these animals are so close, we're more likely to have two viral strains coming in. They can actually recombine within the cells. So viruses move into the cell and within the cell start replicating. If two viruses infect the same cell, the genetic material of these viruses can be exchanged and make what we would call the next superbug. So they can essentially exchange genetic information, almost like sexual reproduction and how we exchange genetic information. Viruses have this ability to exchange their information and create novel viruses, new viruses we've never seen before. They can be extremely deadly. And this is what we've seen before in pandemics from swine flus. Okay. When we talk about infectious agents, we have to see how do they get into the body. Well, different microorganisms have different routes, whether that be viral, whether that be bacterial. We have the fecal oral route, which is one of the most common routes. And this has to do with the GI tract, the gastrointestinal tract. So as we consume food, we're introducing bacteria and viruses to our body, and then they leave through the gastrointestinal tract, through the colon. As these microorganisms reproduce in our body and leave through our colon, fecal matter actually will contain a high number of these microorganisms, which, if we don't have proper sanitization, if we don't have proper technique of washing our hands, and someone is preparing food, it can go back from the fecal route into the oral route and contaminate people. Our skin can also be a portal of entry. This is the epithelium that we have on our skin. Any kind of abrasion or scratches that we have in our skin can be an entry point for microorganisms. Looking at our urogenital trackways, these are the mucosa of these trackways. We have specific microorganisms that can adhere to these areas. So microorganisms that cause urinary tract infections adhere to the mucosa, to the inner lining of the urethra, and they can stick there and start replicating and cause a urinary tract infection. Looking at the peritoneal portal, this would be breaks in the skin again into the peritoneal area. So that would be filling up an area with bacteria. We also have entry via the eye. So any kind of conjunctivitis. Conjunctivitis can be caused by a virus, by bacteria, by fungal. It's also allergic conjunctivitis as well. So entry into the eye, as we see. Okay. Looking at the portals of the body, again, different microorganisms take different routes. So depending on the type of microorganism, let's say if something like HIV landed on your skin and there's no abrasion, no cut, well, then it's really going to be innate in that sense. It would not be able to mount an infection. But if HIV entered into the vascular system, this is where it would be able to mount an infection. Same thing for respiratory diseases, and this is why people wear masks to cover up their nose and their mouth to prevent viruses like from N95 masks and bacteria from moving into the respiratory pathways. Looking at biosafety itself, there's a lot of really important things that we can do. So whether you are interested in a clinical setting or working in a research laboratory, hand hygiene is going to be important no matter what. It applies to all of us. This is why we see that sign at restaurants in the bathroom that says employees must wash their hands. This is preventing the transmission of disease from the fecal oral route as people are handling food following using the restroom. We also have what we call PPE. That's our personal protective wear. You can see this man on the right is wearing his PPE, a kind of more of a hazmat suit in this scenario. PPE has many different biosafety levels depending on the environment you're in. PPE may just be a mask or it may be a complete contamination suit. When we talk about respiratory hygiene, coughing hygiene, that would be like coughing into our elbow instead of projecting our cough out or sneeze outside. As far as maybe the nursing practice, we have safe injection practices. We know we never want to recap needles. Many nurses have actually accidentally stabbed themselves from recapping needles or dropping needles and improper ways of picking them up, or improper ways of disposing of needles. So safe injection practices are extremely important. That goes along with how to handle sharps. We also have patient isolation precautions. So we have places that we isolate patients depending on the transmission cycle of disease. Then we have sterilizing instruments and devices, which we know is extremely important in a medical environment, especially in a surgical environment, to not pass disease from one person to another. And also cleaning and disinfecting the environment around us to not spread disease. Looking at biosafety levels, we have BSL. It's a biosafety level 1 through 4. Biosafety level 1 is the least risk, and we're going to be looking at infectious agents within these biohazardous situations. So these are just basic precautions in biosafety level 1. So that could be handling microorganisms in that laboratory. Biosafety level 2 is highlighted in yellow here, is what we see most teaching laboratories to be. Almost all of the organisms fall in BSL level 1, but BSL level 2 also contains microorganisms like Staphylococcus aureus. Staphylococcus aureus can cause infections, especially in someone who maybe is immunocompromised, but it's also an organism that we can find naturally on the human body as well, but that our own microbiome, those microbes that are covering our body, can protect us from and keep their numbers in check. So biosafety level 2 has barrier precautions. So depending on the laboratory, this could range from goggles to laboratory coat to mask, just depending on what we're looking at. Biosafety level 3 is when we're working with human pathogens that can cause severe illness. And that you have to work in a biosafety cabinet, a BSC, in these situations. So the microorganisms are underneath a hood and locked. The difference between BSL-3 and BSL-4 is really the virulence of the pathogens. So these are more lethal pathogens. And BSL level 4, there's no cure. So if someone was to accidentally infect themselves in the laboratory, there would be no cure for BSL level 4 disease. Okay. We talk about our health and lifestyle. It shows how we have the ability to contract disease, these infectious diseases. So age is a big risk factor for infectious disease. So young is categorized as under 3, old is categorized over 60. At this point, our immune system is not operating the same way. For the very young, the immune system is not fully developed at this point. And for the very old, the immune system is starting to break down and lose some of its function. So people are at risk who are young and old to infectious disease. We also have the host's genetic makeup. So based off your cell type, microorganisms which have a specific portal of entry are going to target specific tissues, whether that be mucosa, whether that be epidermal tissues. They're going to have a specific tissue type of interest. And that tissue type, if we were to zoom in and look at the membrane of that tissue type, is going to have receptors that these either bacteria or virus bind to. So if your genetic makeup doesn't have that protein, that receptor in which a bacteria or virus binds to, you cannot get this disease. Unique situation of this is that they found that naturally born people this was in South America, there was a very, very small population of people who had a genetic abnormality that prevented them from being infected with HIV because they did not have the receptor that HIV interacted with. If you've ever heard of also the genetic modification of humans, there was a researcher in China who was able to do in vitro fertilization and genetically modify two twin girls to lack the receptor, which would enable them to be infected with HIV. Therefore, these two girls were not able to ever become infected with HIV because they lacked the receptor because they had been genetically modified. Okay, we also have host hygiene and behavior. So this is a lot of the washing the hands, using hand sanitizers, which work extremely well if you have access to them and don't have access to washing your hands in a situation. Hand washing as well has to be done correctly, and hands have to be dried in order for it to be effective. So hand sanitizers are a good substitute if you don't have the time or the environment to wash your hands in. Also, as far as behavior, cooking food is really important. A lot of times we're working with raw foods. If we're working with raw foods on a cutting board and then we cook them, we have to remember that that cutting board now has already been inoculated with whatever microorganisms were on the raw food. So even if we fully cook the raw food, the cutting board, if we were to put lettuce on it after, the lettuce could become infected. This is something that can happen with all types of raw foods. So we can have farmlands, agricultural lands where there's runoff from animal agriculture, where manure can end up coming in contact with the agriculture. And this plant agriculture, when consumed, if not cleaned correctly and has water from these manure runoff areas, can cause infection. Maybe E. coli could end up in the food. Okay, sexual activity is another thing that is extremely important that works into lifestyle factors for contracting disease. Sexual activity is going to be one of the factors that we can look at a couple different ways. While there are safe sex practices, abstinence would be the safest sex practice because it would have no issue of any kind of barrier protective method breaking. And also with barrier protective methods such as condoms, they will not cover all of the genital area where there could be an infection. So increased sexual activity or not safe sex can increase our likelihood of contracting an infectious disease. Looking at nutrition and exercise, it's important that we stay healthy and keep our bodies healthy through exercise because our immune systems will regulate disease coming in. So staying healthy increases the ability of our immune system to regulate incoming foreign invaders. Looking at lifestyles and infectious disease, we can talk about a couple things like pre-existing conditions. So pre-existing conditions can make you more susceptible to disease. So any kind of autoimmune disorders, chronic infections, or even if you have cancer, or lupus, these things will make you more susceptible because they're compromising your immune system to disease coming in. Also, our occupation, if we're looking at people in the allied health science, if we have exposure to people with infections, if we're working in hospital settings, we have high exposure rates to people. Even laboratory personnel, if we're working in research, we have high exposure. Agriculture workers, people working with animals, are going to get high exposure rates too. A lot of those pandemic like bird flus, swine flus that come out, it's the agricultural workers that are first exposed to these diseases. And a lot of times agricultural workers may not be in the situation where they're able to receive healthcare in these environments. Looking at hunters as well, as hunters go into environments where there's not that many humans, as they move into forests, they move into areas where there is higher disease, they're more likely to contract disease. Also, if we look at something like HIV, the way HIV got into the human population is through hunting. So it was in monkeys. It didn't cause the same signs and symptoms that we see. But when the monkeys were butchered, the butcher probably had a cut, maybe on their hand from using large knives. And when the blood from the monkey entered the blood of the human, the disease was transmitted into humans, and that was our patient zero. Looking at prostitutes as well, or sex workers could have a higher risk of exposure if there's physical contact between people. Looking at our immune system and pathogenesis, we know that our immune cells can also cause damage to us. So after infection, we have sequelae, which can cause damage as well, even in healthy hosts. Let's talk about emerging disease and re-emerging disease. So emerging disease is something that has just been seen by the population that is relatively new to the population. On the other hand, re-emerging disease is something the population sees kind of these natural ebb and flows of disease going up and back down slowly in the population. When we look at disease, re-emerging diseases and emerging diseases, we can look into deforestation and see an association with deforestation and disease. Here we see deforestation, and here we see disease on the right with a specific microorganism. As people venture into these forests, into these areas we haven't been to before, we get exposed to novel diseases, new diseases we've never seen, and we bring them back into the human population. Okay. So now we move to the point where we have our questions at the end of the lecture. This is very similar, question 1, to what we saw before. So if you didn't catch it the first time around, this is a great time to go over it. Question 1 is based on the information in the graph below, which organism is more virulent? Again, I'll give you a second to pause here before we go over the answers. Okay, it's going to be agent 1, which is more virulent. We see its LD, lethal dose at percent mortality, 50, reaches here at only about 400 microorganisms, where the LD for agent 2 is 600, meaning you need a lot more microorganisms to cause an infection and to cause mortality rates in agent 2 than you do in the LD 400, LD 50 at 400 microorganisms for agent 1. So agent 1 is more virulent because you need less microorganisms to cause mortality. Okay, question 2. Mahadu comes to a clinic experience of fever, rash, and high blood pressure. He is experiencing, again, this would be your time to pause. We'll move on to the answer. These would be signs. So fever is something we can quantify. We can take somebody's temperature. A rash is something we can see. High blood pressure is also another thing we can quantify by taking their blood pressure. So that would be an example of signs. Question 3, an epidemic is? The answer is an increase in the number of cases of a disease in the community. Question 4, determine the biosafety level of the following pathogen. Agent A can cause severe disease, including death in some patients. It can cause disease by either inhalation or ingestion. It can be treated with a readily available antibiotic, doxycycline. This is a level 3 biosafety. And that's because we have the antibiotic. So even though it can be deadly, you might think it's biosafety level 4 because it has a readily available antibiotic. It brings it back down to level 3. Question number 5, according to the following graph. And the answer to this question is rates of malaria increase with forest loss. So here we can see on the x-axis, we see forest loss, so increase in forest loss. On the y-axis, we see malaria. So we see a direct correlation between the two. As forest loss increases, so do malaria cases increase. So according to the following graph, rates of malaria increase as we lose forest. Okay. Again, this is where we hit your study guide with section objectives. So here we can see these sections. These are questions that you can start answering and going through the lecture. It's not expected that hearing a lecture once, you're going to all of a sudden absorb all of the information. So typically listening to a lecture twice or taking notes or reviewing these as you go through the lecture to keep track of them is a good idea. Okay, that concludes the second lecture.